really happy to be here to have the chance to share with you a bit about what's going on microscopically at Chautauqua Lake. Some things that you probably know a lot about harmful algal blooms and what happens when things get out of control. But one of my goals for today is to really share with you the value that the algae provide to us as a whole community at the base of the food web. So we'll go through a few different stories today. But what I always like to start off with is what do I mean when I say algae? Essentially, these are microscopic aquatic plants. They don't have roots or vascular systems like land plants do, but they perform a similar ecological role at the base of the food web. And so this foundation is really where all the energy comes in to the system. Okay, so algae are the entry point for energy and sugars that they create through photosynthesis. And so they take energy from the sun and use it to build carbons into sugars. These are some samples from a trip we just did a couple weeks ago with the Alliance, some water samples from the south basin of Chautauqua Lake. So these algae were just collected a couple weeks ago. And so you can see just by looking at them that there's some different sizes and shapes and colors, and we'll talk about that in just a moment as well. But two questions I frequently get asked when I'm out in the field is one, how are the fish? I don't study. And number two, when are we going to get rid of all the algae? So as an algal ecologist, I hope you leave today understanding that there are really important good things about having algae in the system. In large part, especially if you're an angler, is that if we have no algae, we have no world-class fisheries here. So algae are the entry point for energy. They feed the zooplankton or the animal plankton, and that feeds fish, which feeds bigger fish, which feeds the big monsters, right? So we need to have a functional, healthy algae community here. And so that's what I want to share a little bit more about with you today. What does it look like when we take an algae sample? This is what my students get to look at, right, guys? I have some of my students are right here. I get to enjoy this. What you notice here in the sample from Chautauqua Lake is already a huge diversity, right? Different sizes. Some of these algae are living singly. Some are living in colonies and chains. Some are very large and long. Some are small and round. There's different coloration patterns, and so right away we have a good sense that there's a lot going on that we don't really have the opportunity often to pay attention to below the surface of the water. Algae diversity is really incredible. From the best numbers I could find, and these are old, but these are the best ones I could find, the estimated number of algae species in aquatic environments around the world is somewhere between 15 to 45,000 different species, and this number is probably considerably higher now, especially as we have advances in genetic analysis and instrumentation. We're learning and identifying new things all the time. So that number is probably considerably higher at this point. One of the questions that we often ask, and we've been asking for a while, is why are there so many different species of algae? One of the foundational concepts in ecology is the idea of competitive exclusion. That means that if you put two species in a confined area with not a lot of resources to go around, they're going to be in competition with each other. They're going to be fighting for those same resources. And at some point, someone's going to win. Most of the time, right? And so that means that you have the exclusion of other species and one dominant group that's there. But we do not see this, right, at Chautauqua Lake. We don't see it in most freshwater systems. And so why doesn't one species win? This is called the paradox of the plankton, a term coined formally by G.E. Hutchinson in 1961, looking at the fact that we have so many hundreds, if not thousands, of species living in the same place at the same time. So how does that happen? Why do we have that? And the answer is pretty clear once we take a bit of a step back and realize how dynamic aquatic habitats are. They are constantly in a state of flux. There's rarely a moment where things are stable for very long. And so with all of these differences in streams and lakes and ponds and oceans and wetlands and their interconnectedness, there's a lot of physical habitat diversity that's available for algae to occupy. And so even within a single system, take Chautauqua Lake, for example, there are changes in space and time. So what's happening, the environmental conditions at the shoreline of the lake are very different than what you might see in the open water habitat, or even from the surface of the water down to the deeper, darker, cooler depths, right? And so even within a single lake, you can see a lot of physical, spatial differences. Okay. We also see differences in time. So we have changes that happen over the course of a season. So what's happening in the lake in January with ice cover is very different than what you might see in May versus July. Think about the lake in October and November. And so these changes, even over the course of 1 year, create opportunities for many different species to grow. This gets compounded as we look at year-to-year variation, and I'll show you some data later that look at the longer-term sea slap record and just show you how much we can have differences from 1 year to the next. It's never been the same in the 5 years I've been here looking at the lake, so it's fun to see what's from a scientific perspective, it's fun for me to see what's happening each year. But what this boils down to is all of this environmental variation means there is a highly diverse group of organisms at the base of the food web. So this is what the algae are doing for us. There's a lot of different things that are there, not just the harmful species that we'll talk about in a bit. And so we have lots of different growth forms. Some algae like to live free floating in the water column. Some regulate their buoyancy to be in certain places in the water column. Others live attached to rocks or plants or even other algae. And so you see a lot of different growth forms, different entire taxonomic groups, different classifications of organisms, right? In this umbrella term we call algae. And each of those species are variable in their ecology. They have their unique habitat preferences, their resource requirements, and even some variations in their metabolism. So I'll tell you an interesting sidebar that most algae are photosynthetic, right? They make their own food, but there are some species of algae that can switch and actually eat bacteria instead, depending on the conditions, right? And so there's always an exception to the rule in ecology, which keeps it interesting, but also very challenging. So the diversity in the species is because of all these different resource opportunities. So I'm not going to do a taxonomy talk where I talk about all these different groups today. I won't melt your brains, but I do want to just highlight these are the major categories of algae that are present in aquatic environments. Some of them are found exclusively in marine systems, others are found exclusively in freshwaters like Chautauqua, but many cross over and have representatives in multiple kinds of habitats. I'm only going to focus on the top 4 today, but I'd always be happy to provide additional information if you have questions. These are 4 of the ones that are common in the samples that we collect at Chautauqua Lake, so they're representatives in these from these groups. Most of the year, actually, we see a lot of these different species present. And so I'm just going to hit a few highlights, and I'm going to talk more about this first one, cyanobacteria, in a moment, but just to show you what they look like and how they're different, a little bit about their ecology. So cyanobacteria are different than all the other ones. They're a whole different class of organisms called prokaryotes. They have a very different internal cell structure compared to all the other algae groups. So cyanobacteria are bacteria. They tend to be smaller in cell size. They don't have any they've limited internal organelles within their cells, but they are one of the most ancient groups of organisms. They've been around for estimates range 2.5 to 3 billion years on our planet. So they've been around a long time, and they're very good at doing their job in a lot of different environments. We'll talk more about why that is and how they can get around some of the other things that other groups of algae can't. But they're fascinating to look at, and even in this picture, there's 2 or 3 different kinds, just you can see in the different shapes present here. These are all samples from Chautauqua as well. Another group are the dinoflagellates. This is Serratium. These dinoflagellates, this particular group has these armored plates. But the other thing I wanted to point out is that the shape of this, which kind of looks like the Eiffel Tower, at least that's how I tell my students to look for it. But Serratium has this sort of different shape because, number one, it helps slow them down from sinking out of the water column. So we see algae in different morphology so that they can stay afloat or have a high drag. Other times they have longer spines towards the end of the season to help prevent them from being eaten by something else. So algae have a lot of different physical characteristics as well. The dinoflagellates are also really good swimmers in the algae world. Green algae are, this is Pediastrum. This is, you can see that the coloration, these are not preserved samples. These are just straight up from the water. You can see that the Pediastrum has a more bright green color. So these are a highly diverse group. This is just one example that's here. But again, all the algae have chlorophyll A as the primary pigment that does photosynthesis, but everybody has their own little fingerprint of pigments, right? Each has their own little package of different kinds of pigments, which give them unique coloration sometimes, even when you're looking at them on the microscope. Diatoms are my personal favorite. These are a wonderful group of golden algae that have a glass cell wall. They have, each species has its own ornamentation and markings, lines and stripes and sizes and shapes. And these glass cells will, once the algae are done growing, will sink to the bottom of a lake and often become preserved as fossils. So if we understand the diatom ecology today and what drives their growth today, we now have a record of environmental change going back in time, hundreds, thousands of years, millions of years in some cases for the deeper rift lakes in Africa. So they're particularly beautiful to look at, but also very handy from a number of scientific perspectives. Some of the factors that control algae growth are going to vary depending on the species, but the big players are light, temperature, and nutrients. These are the types of things that really affect the rate of photosynthesis, which affect algae growth. I do want to point out here that nutrients include nitrogen and phosphorus. A lot of times we talk exclusively about phosphorus, and there's good reasons why we do that. But we did some experiments in 2015 that show that nitrogen and even different forms of nitrogen can stimulate algae growth as much, if not more, than phosphorus in certain cases. There's also some interesting work looking at harmful algal blooms and how nitrogen might affect the toxicity, if not the overall growth rate, but their ability to produce toxins. So it's important that as we're looking at nutrients that we're not forgetting the nitrogen side of the story. Lake mixing is another important piece that sort of ties together the physical environment, the light, temperature, and nutrients together. But if you've ever been swimming and you are in the warm waters of the surface and all of a sudden you feel your toes get cold, everybody experienced that probably at some point. So that's actually these thermal layers that are occurring within the lake in the summertime. And those layers typically don't mix in many systems. They stay separated, which is why you can have really cold water down below and warm water on top. But the mixing pattern, that physical condition, alters light, temperature, and nutrients. There's certainly a whole suite of other chemistry that we could talk about, pH, salinity, trace metals. I'm not going to go into that in a lot of detail, but there are a lot of different things that we pay attention to depending on the system that we're interested in and some of the environmental conditions that are happening. These are all what we would consider abiotic or environmental factors that affect algae growth. But I do also think it's important to point out that algae are a living part of a living system where we have interactions with other members of the algae level in the food web, where they're in competition with each other, fighting for those resources. Additionally, there's also the potential for being affected by the next level up in the food web, so grazing or removal by zooplankton, the animal plankton that would feed on algae. So they're affected by the environment, but also they're interacting with the other biota that are present in the lake. So I want to show you what this looks like, what algae growth can look like over the course of a season. But I want to preface it by saying that you actually all probably understand this concept. You just maybe haven't thought about it in terms of what's happening below the lake's surface. If you've ever planned landscaping or you've ever looked for the flowers blooming in the spring, you know that certain plants in the terrestrial environment bloom at a certain time, right? Other ones grow early, some grow later. If you're planning your garden, you're going to want to have greenery for backdrop, and you're going to time the bulbs that you plant so that things emerge at particular points in the season. And so you understand that planning, that sequence of events, right? The same thing that we see happening on land is happening under the water with the algae community. So I'm going to show you this here. So it's interesting to look at the changes over the course of a season. This is a classic figure from Wetzel or one of our premier limnology textbooks. But what it shows you is an example from Lake Erkin in Sweden, which is a similar pattern that we see for lakes around here as well. Okay, so this is an example lake, but it's the pattern is pretty consistent. We start in January here, and we move through the course of the season, through the summertime and to December, looking at what's happening month to month. And then this looks at the algae biomass, in particular phytoplankton, which are the open water algae species. Okay, so not the stuff growing along the shorelines or attached to the bottom. But the algae biomass here, you can see over the course of the season, once the snow and ice comes off in the early spring, we have differences in total growth. Right? So there are peaks and valleys, there's ebbs and flows to the total amount of algae that are growing in a lake at a particular time. In addition to changes in those peaks, which we tend to see in the early season and the late season, there are different groups of algae that are dominant at particular times. Okay, so just like you would see the crocuses and the daffodils early season, right? And then you have to wait to see your peonies and your other types of plants blooming later. We see the same sort of succession in algae species. So the diatoms tend to like these shoulder seasons where the lake is mixing. They do really well in the early spring and in the late fall. But you can see that the different groups change, and this in particular I'll draw your attention to are the cyanobacteria that normally really like to grow well in July and August in most systems. So again, the takeaway here is that change is an inherent part of looking at the algae community. One sample doesn't tell you everything. Things are in a state of flux, and there's a lot of diversity to explore. I always tell my students that it's important to know how you get your data. So looking at sampling approaches and plans, but also the techniques that you're using to collect information dictate how you can interpret those results. So there are a million ways that you can measure algae. I won't talk about all of them. You can come take methods with me in the fall if you're interested. I use this example with my students. But each approach tells a slightly different story, right? And so the most common ways that we look at algae for most people that are doing this kind of research are to actually take a sample and look at it on the microscope and identify the cells to species and count how many are there. Right, guys? Casey's shaking his head, no. But yeah, so it's very labor intensive and requires intense training, right? But it's the best way we can look at the number of cells and the species makeup, right? Who's doing the growing at any given point. Oftentimes we're not so interested in the species diversity, but more the total biomass. How much is there? How much production is happening? And we can do this by looking at chlorophyll A as a pigment of photosynthesis that correlates to how much algae are there. More pigment, more algae, okay? Oftentimes we'll take lake water and we'll filter it down to a more concentrated sample, and then we'll extract that pigment and break it up, and we'll extract the pigment and read it in the lab, and we can see how much material is there for the pigment. There's also some techniques that use fluorescence, so using the whole live cells and looking at these on a fluorometer in the lab. But we're doing some really cool things with remote sensing, using satellites and drones now to look at the fluorescence from farther away. So not actually looking at it in the water sample, but from a distance. These are the most common things that you'll see in terms of people tracking algae growth through time. What this really boils down to is looking at productivity measurements. And when we say productivity, we're interested in how much primary production or photosynthesis is happening, right? So high productivity means there's a lot of plant and algae growth in a lake. So the classic ways that we measure that are to use the extracted chlorophyll data, but also we use things that are other proxies, like total phosphorus. Phosphorus tends to correlate tightly to algae growth, and it's a little bit different data analysis and processing, and so we track total phosphorus, and we also look at water clarity using a Secchi disk. If you haven't people, you've used Secchi in here? You guys know what a Secchi disk is? Okay, some of you. It's a super high-tech piece of equipment. It's a black and white disk, right, that you lower over the side of the boat and you wait for it to disappear. And that point in the water column where it disappears and your eyes can no longer see it is about 10% of the light remaining. And so it's a bit subjective from observer to observer, but it's super easy to train people, and it's an inexpensive tool, and you can have lots of these out with lots of different people. So the idea is that looking at water clarity, as your water clarity declines, as it's less clear, you have more stuff living in the water, so it correlates to algae growth as well. There's some caveats to that, but by and large, it's a really fantastic tool. And so we have long-term records that allow us to look back decades before we had all these high-tech instruments that we have today. So some states, different states, New York State doesn't seem to do this quite as much. I'm still learning. But some states will actually combine these three productivity measurements into what's called a trophic state index, or a TSI. So if you see that, if you're looking at examples from other states and you see that, the thing is that each of these tells a slightly different story, and so when we classify lakes or categorize them as being highly productive or low productivity, you sometimes get different classifications depending on what you're looking at. So sometimes we'll fold these all together into an index. So if you're looking at how you measure algae, I encourage you to dig a bit deeper and understand how those data were collected. So that's the first part about algae, Algae 101. What does this mean for water clarity going forward? Again, please remember, I hope if you leave today that you remember this, that algae are a normal, healthy part of lake ecosystems. We don't want to get rid of all the algae. That doesn't mean we don't have problems when they grow too fast. So when we have these imbalances and this overgrowth of algae, we can have harmful algal blooms with implications for water quality. In freshwater systems, this is primarily The fault of the culprit is typically the cyanobacteria, that first group I talked about. It's a little bit different actually if you look at harmful blooms in marine systems. Those are actually red tides and those are dinoflagellates, so it's a whole different group because it's a different environment. But the dinoflagellates in Chautauqua are non-harmful, so don't worry about it. But cyanobacteria are one that we want to pay attention to. Some of the things that can happen with respect to water quality are changes in transparency. We can get this sort of pea soup cloudy appearance here. Aesthetics, changes in color, odor, and appearance. And one of the biggest things, I'll talk a bit more about this, but looking at the toxins that can be produced by cyanobacteria can create issues with drinking water processing, beach closures, recreational access, and all of the economic implications that come from that. Once the bloom is done, that's not the end of the story, unfortunately. That biomass gets decayed, gets decomposed, and that can use up oxygen as other bacteria process it. And so we can have post-bloom oxygen depletion, which may create the opportunity for a fish kill, since fish rely on oxygen in a well-oxygenated water column. Anytime we're messing with the base of the food web, we could have, for long periods of time, we could also have changes in the food web structure overall, in particular because cyanobacteria are not terribly easy to eat or nutritious for other parts of the food web. And so when we start changing how that energy is coming into the system, we have potential to alter the rest of the food web as well. I'll refer you here, the New York State has excellent resources on harmful algal blooms. So a lot of the stuff I'll pull in the next few slides are from resources that are freely available through the Google machine. So I encourage you to take a look at that. I'll have a slide with some links up at the end, but all these are really easy to find. So great to have excellent resources from the state. Interesting that different states define an algae bloom in slightly different ways. So what happens in New York State is if a visible bloom is seen, so something like this, these are images from the HABS brochure. So you can see that there's different ways that algae blooms might appear in the water column. Okay, so if you see anything like this, a sample is collected and then it's sent to the lab for analysis and confirmation. In that lab sample, at least one of these criteria has to be met for it to be a confirmed bloom. We need to measure the blue-green chlorophyll, or a pigment that's unique to blue-green algae, those cyanobacteria, has to be above 25 micrograms per liter, or microscopic confirmation that they are cyanobacteria in that bloom, or microcystin toxins. One of the toxins that they produce has to be measured at a certain level. It can also be classified, there's another flow chart if the microcystin toxins are higher, it can be classified as a highly toxic bloom. So one of these criteria need to be met, and then it's called a bloom officially in New York State. The causes of HABs, as we heard from our keynote, really looking at excess nutrients, so more nitrogen and phosphorus coming into the system combined with warmer temperatures. If you remember the seasonal succession, those cyanobacteria already love to grow in July and August. That's their wheelhouse, to grow in those warm temperatures, right? So we're exacerbating the temperature issue through climate change as we're increasing our surface temperatures of lakes around the world, and definitely we're seeing that across the Northeast. But also as we combine that with excess nutrients, then we have the blooms that get triggered. As part of Governor Cuomo's HAB initiative, there was an action plan that was created for the 12 priority lakes. The Chautauqua Lake is one of those. I encourage you to look at the HAB action plan that's available. There's fantastic information in there, so I'm drawing a bit from that document here. There was an assessment of risk factors for New York State lakes for HABs. There were 4 factors that were identified: high phosphorus levels, having invasive mussel species, so the zebra and the quagga mussels, the length of the lake, and then the orientation of the lake, the compass orientation of it, the way it's positioned. Chautauqua, we have 3 of those 4 risk factors. So we have a high risk for algal blooms, as you've many experienced and are here to learn about. So these are just some images of cyanobacteria that were taken from Chautauqua Lake at various points. As I mentioned, cyanobacteria are an ancient group. They're very good at their job at living in different locations, and one of the things that makes them so good at this is that they're able to make their own usable nitrogen. So you may not know, but you're all right now breathing about 78% nitrogen gas, N2 gas in the atmosphere. It's 2 nitrogen atoms that are bounded together by a really intense triple bond. You breathe in that nitrogen gas and it goes right back out. It does not interact in your body, right? It comes in and it goes out, it comes in, goes out. You'll never notice it. The problem is if you need to get your nitrogen, right, you have to have something that you can use, right? Algae needs something that they can use, ammonium or nitrate, and so they can't access or use N2 gas that's in the atmosphere, except for cyanobacteria. They have developed these specialized cells called heterocysts that allow them to actually take that N2 gas and split it and reorganize it into a usable form right inside the specialized cell. To tell you how cool this is that cyanobacteria do this, the only other way that this happens naturally is through lightning strikes. That's how much energy it takes to break apart that triple bond, and these cyanobacteria do it all day long in their heterocysts. Yeah? Cool, right? So they have this capacity to do nitrogen fixation. We do it now industrially through the Haber-Bosch process to create industrial fertilizers, but it's been happening in the natural world through cyanobacteria processing on land and in the aquatic environment. Anyway, the nitrogen fixation allows them to, if they have access to phosphorus, then you're alleviating one of the big nutrient restrictions, right? So they can make their own nitrogen, give them a little bit of phosphorus, and they're off to the races. So they're very competitive in the late season when there aren't a lot of nutrients freely available because they can make their own nitrogen. Unfortunately, these good growers in the late season are also able to produce toxins. So liver toxins, neurotoxins, a whole suite of different chemicals. And it's tricky. We don't understand everything about them. But these toxins are a public health concern because if you have contact with them on your skin or through ingestion, you can have some serious side effects. The outcomes range from an extremely itchy rash, which I can testify is very uncomfortable but not life-threatening, from exposure to a bloom, all the way up through neurological deficits. And there's been documented cases of livestock and pets that have died from exposure and ingestion of cyanobacteria blooms that were toxic. So it's really important that we pay attention to that. Part of it is because like dogs will go out and they'll accumulate all of that material on their fur. They'll come out of the water and then they'll lick themselves clean and they ingest all of that really concentrated. So that's why we have issues with pets and livestock with toxins. So these are some of the causes and the culprits for HABs. I do want to mention just quickly a little bit about the lake water quality records that we have. And so this is going to look over the course of a season from 2018. These are available on the NICE-FOLA website. You can look up the CSLAP reports that are there. So we are incredibly fortunate to have a dedicated team of volunteers that go out and collect these samples and have been doing it for a long time. And then also to have these data publicly available through NICE-FOLA is pretty incredible. So you can go on and learn and educate yourself about what's happening year to year. And then I'll also show you some long-term perspective data. So I know this is a little bit small, probably for the people in back, but what I really want you to take away from this is these are the open water samples from 2018. And what you'll notice over the course of the year, this is chlorophyll, and this is starting in mid-June, you'll notice that we have an increase in chlorophyll till about September 3rd, right? And then it drops back off. The blue bars indicate the cyanobacteria, but what you'll notice is that even when the cyanobacteria are at their worst at Chautauqua, it's only maybe a little over 50% of the community. There's a lot of other stuff that's still growing at the same time as the cyanobacteria. And eventually, they lose that competitive edge and the production drops off as we get into the cooler nights and the shorter days in the fall. The story is a bit different when we look at the shoreline blooms. You'll notice that these bars jump a lot more dramatically. And so when there's a bloom, the production is really high, the chlorophyll is really high. You'll also notice that most of the bloom that's collected are cyanobacteria. Right? So this could be, we don't really understand this necessarily yet. This is some stuff that we're working on, but it could be that they're growing more in these places, but it also could be that they're being physically transported and accumulating along the shorelines. So in the Lake Chautauqua that's very large, we have a lot of work to do to understand the origination of blooms and their transport and accumulation. So that's some of the stuff that we're working on right now. Anyway, so differences in terms of where you collect the data. So the spatial difference from the middle of the lake to the shoreline is really important that we're paying attention to that as we try to understand algae growth. Also to point out that the CSLAP program does measure toxin levels. And so this lower line is the drinking water threshold, and then this is the swimming criteria. So there are many points throughout the summer where we're having high toxin levels. So when you're above that threshold, you'll see the beach closures, and that's why. In 2018, Chautauqua spent 18 weeks on New York State's HAB notification list. So we know that this is an ongoing problem, it's affecting a lot of people in many different ways. So this is what's happening over the course of 1 year. I want to take a moment and show you those productivity measurements for the clarity with the Secchi disc, chlorophyll, and total phosphorus over the long haul, right? So when you look at what's going back to 1986, 1987 with the Secchi disc and the water clarity here, the deeper the Secchi disc, right farther down, the clearer the water, and this would be classified as low productivity or oligotrophic. You can see that Chautauqua, we hang around on this borderline of meso to eutrophic, mesotrophic middle production, eutrophic highly productive. And so you can see that we sort of hover around this threshold, but there's not a long-term pattern that's really evident here in the open water samples. If we look at chlorophyll, we have the record for this as well. You can see at the chlorophyll classification, we're more in the eutrophic zone, okay? Again, variability, but not a clear long-term trend from these samples. The one that's concerning is the increase in surface phosphorus levels, which were solidly in the eutrophic zone, and we are seeing an increasing trend. Again, the methodology and the collection of these data are important because while we don't see clear trends in the bloom frequency in the open water, it doesn't tell the whole story, right? So we have this great time perspective from one place, but in ecology, there's always trade-offs in terms of our approach. You can't sample everything all the time. We have to pay attention to our sampling programs and think about how we can augment all this great work that's being done and try to find ways to help answer some of their questions we have. So the CSLAP data, it's remarkable. We have a fantastic data set for Chautauqua and other lakes all around the state. So it's as a lake ecologist, it's really fun to work here with lots of info. If you see a bloom, I will tell you I am a science type doctor, Ph.D. doctor, not a medical doctor. So I refer you to, for safety purposes and liability, I refer you to the DEC harmful algal blooms policy, which is know it, avoid it, report it. So please pay attention to what's happening in the water conditions when you're in and around the lake. Look at the bloom alerts from the DEC. You can sign up to get updates from them. And there's a new NYHAB's mapping program that shows the bloom alerts as well this year, which is very exciting. So know what you're looking for. If you see it, avoid it. You unfortunately cannot look at a bloom and know if it's dangerous. You can't, right? The only way we can confirm toxins is either if you get one of those lovely rashes or if we send it to the lab to look at for samples, okay? So it's not worth the risk. So stay out of the water if there's a posted sign or if you see a scum. If you do have contact with blue-green algae, it's really a good idea to rinse with clean water and keep any eyes out for medical concerns and seek medical attention if necessary. You can also be a part of the solution by reporting suspicious blooms to the DEC. Knowing what you're looking for, there's an easy way to report those via the website. Once the DEC gets notification of a suspected bloom, they follow up and we collect samples. Volunteers go out and collect samples to send those into the lab for confirmation, okay? There's a lot of times we get these questions about what we should do. We've talked about them a bit in other sessions, so I'll briefly just say that I'm really not on the management end. I'm really more on the algae research end. As you can see how excited I get about cyanobacteria doing nitrogen fixation, right? I'm not on the management end as much, but I do think it's really important that people pay attention to what are the sources of these, so and what are some things that you can do. So the HAB action plan is part of that HAB initiative is available, and I would encourage you again to look there. There are always long-term and short-term solutions when we're looking at resource management, and the analogy that's often used is treating a disease versus treating symptoms. You often need symptom relief, right? You need that Band-Aid if you get a cut on your arm, but you also need a doctor's attention to fix the problem and get you stitched back together. And so there's this balance of what we're doing to our natural resources in a way that helps people, but also keeps the system healthy in the long term. In the HAB action plan, most of the recommendations really focus on watershed management, so reducing the sources of those nutrients that are coming in. This is considered the long-term treating the disease, right? The bonus here is that it not only helps reduce algae growth, but also helps reduce plant growth, right? So it's a win-win situation. So there's all these ways that we can invest in making our watersheds healthier for reducing nutrients coming into the system. There's also in-lake treatments that we can do, these short-term Band-Aid solutions with variable success, with the caveat they also require DEC permit in some cases. So I refer you to the HABS program guide, table 6.1, if you're curious about learning more about any of these specific categories. But there's biological controls, chemical controls, physical controls, and some combination of short-term and long-term management is likely to lead to the the most relief in the short term, but also long-term successes. We are still learning. We have a lot to learn about what's happening at Chautauqua Lake, and so I'm fortunate to work with great people around the area and to have fantastic students in my lab. And so these are just a couple of things. If you see us out and about, feel free to say hi and wave at us this summer. You may, in the North Basin, see CHAMP, the Chautauqua Aquatic Monitoring Program. CHAMP is a yellow buoy about 3 feet high with red markers on the side. If you see it, feel free to wave, but please don't hit it with your boat. It's over across from the Lake Chautauqua Lutheran Camp, who's our upland sponsor. And we get a lot of support from local groups. But basically what CHAMP does is it takes near real-time measurements of what's happening with the water chemistry in the lake and then radios it back to us so I can call into the buoy and see what the temperature and dissolved oxygen and pH are at any time. So it's a great tool for us to look at that temporal perspective, right? Like really shortening down the time frame between samples. And so we have more detail work that we can do in the 2-week CSLAP samples, but we can get this faster and tell a little bit more about what's going on. We also have the new project that's sponsored by Corning, looking at HAB surveillance with drones and hyperspectral cameras. So this is really exciting. This is just getting off the ground. Haha, nice drone pun for you. And then we have a temperature profile survey that my students are working on this summer, looking at those temperature layers, the stratification in the lake, what's happening spatially with that, and can we use that to understand more about nutrients and link it to algae growth as well. So lots of exciting stuff this summer. In summary, algae are a normal, healthy part of Chautauqua Lake. There are water quality implications if things get out of hand. We have frequent and persistent blooms here, but there's a lot that we can do to reduce nutrient inputs. It's challenging, but we know the things that we should be doing. So it's a matter of getting everybody pointed in the same direction and working together to conserve this great resource. If you're interested, there are data and resources here. Many people to thank in the community and at Fredonia and our teams of science. That doesn't happen in its own vacuum. We all work together. It's a great collaboration opportunity in the aquatic world. And with that, I'll